I am Abdessamad Zerrouqi, adjunct at the Biochemistry Department of Medical University of Warsaw, principal scientist for a project to develop a strategy to overcome immunosuppression in cancer in collaboration with Dr. Beata Pyrzyńska at the same department. The goal of this project is to restore the cytotoxic activity of immune cells that have infiltrated the tumor to kill cancer cells. And the immune cells found a cocktail of metabolic byproducts that hinder their activity. So we discover active compounds that are capable of recovering or restoring the activity, cytotoxic activity of immune cells once exposed or after exposure to a byproduct that tends to accumulate in tumor microenvironment. The results were good enough to prompt us to study and assess the activity of this compound in vivo with mice having implanted tumors. Before I continue talking about the results, we have to mention that actually cancer management is increasingly relying on immunotherapeutic drugs, which are known as immune checkpoint inhibitors, which remove the brakes in immune system to fight cancer. However, the recent studies have shown that actually these immunotherapeutic drugs have limited efficacy, and only 20 to 30% of patients respond to these drugs, and most of patients don't. So there is a rationale, and there is a rationale for drug makers to try and to push this response rate higher by testing in thousands and maybe in hundreds of clinical trials the combination of these immunotherapeutic drugs with other drugs with different class to achieve better response rate. This is for us a good biological rationale to also combine our molecules, our compounds with immunotherapeutic drugs. And we get a grant from Center of Technologies and Transfer, the Inkubator for Innovation 2.0, to assess the activity of this compound as a single molecules without any combination with immunotherapeutic drugs. The results are as the compounds as a single molecules or single agent have limited efficacy on tumor growth control, and this efficacy fade with time. This lack or this loss of response is common in cancer. We know that tumors are known by developing a resistance, not one, but several mechanisms to evade immunosurveillance and destruction mediated by both innate and adaptive immunity. But also, we, from the result of our experiments, show that actually these compounds probably have an effect on one subset of immune cells, but not all subsets. And therefore, we think that this is rational for combining these compounds with immunotherapeutics that have ability to boost the activity of other immune cell subsets. You may ask what are the benefits that these compounds have in cancer therapy. So the benefit will be gained once these compounds prove their efficacy in combination with other drugs, especially immunotherapeutics. And because the limited efficacy of immunotherapeutic drugs right now and the high incidence of cancer around the globe, We think that chemical alternatives to boost these immunotherapeutic drugs will gain more interest and will be in very high demand. So as the interest for these compounds will be growing day by day, as the immunotherapeutic market will be growing, and it's growing fast, probably reaching $200 billion in the next 5 years. So by using these combinations, we think that we are at the right time to find the right combination of immunotherapeutic drug with our compounds to achieve high cost effective solution and better response rate. I will take the opportunity. Finally, I will take this opportunity to express my gratitude to a Center for Technology Transfer for awarding us with a grant Inkubator for Innovation 2.0, and also I am very thankful for the support of Mrs. Anna Kula and Mrs. Joanna Stepkowska and their teams, and also very grateful for the continuous support of Dr. Pyrzyńska in implementing this project and carry out all experiments. Thank you very much.